So how are you all?
Good, and you, Miss Nandi?
Good. Still doing good.
That's good. I'm glad
Ve-
you're good.
Yeah.
Very good, just staying busy all the time.
Yeah. The situation outside is not so good.
No, it's not,
I've
Miss
been having to do a
Nandi.
lot of orientation stuff because I finally got on with Wellstar. So yes,
Oh, nice. Yeah. Hmm. Today we had meeting. So I heard the number is increasing, but
Yeah,
the number
yeah.
of positive cases are
I know they
increasing.
said that we had 600 new cases across Georgia, but that overall that it has gone down. Yes, there are 600 new cases, but with the way that people have been getting it, it's gone down tremendously. It still
Well,
sucks, but at least we're kind of making
yeah.
progress.
I don't know, but I heard that Georgia Tech, more than 1,000 students tested
Mhm.
positive.
Dang,
Yeah,
And
no, you should look at the numbers at Georgia Southern.
huh,
Those
that's
are
ridiculous.
terrible to look at.
Ooh,
yeah, but I think
Georgia Southern.
USG, they don't have any plan to shut down.
good.
Nope. Uh,
No,
I know
so
that they had said that Georgia Southern was a school that was like built for a face-to-face kind of class, and so
uh-huh.
even when COVID was like at its height, they didn't change the way that they were doing it, and like half the
Mhm.
campus got it.
Oh my
Ooh,
God. Oh
And
dear.
I just moved back up from there, like no joke. This is my first year back in the Atlanta area.
Uh, yeah, let's see how it goes. Really scary.
yeah, Miss Nandi.
Yeah. So especially, our lab, next few labs, it's with microscope and all those things. So
Labster was cool. I like that. I did the simulations. That was kind of fun.
yeah, it's fun, but I'm not sure, how much you are going to learn.
Exactly.
Yeah.
To me, yeah,
I mean,
it's more like
that's fair.
I agree with that. It was fun, but it was like, how much am I really retaining?
exactly.
Yeah.
What
Yeah.
is this?
And it takes long time, like
Oh yes,
yeah,
forever.
yeah, I
And then
know
if you get lost, you're screwed. You don't know. You can't move forward.
Yep.
yeah, like
All about that thing.
I know that one of my student, I think it's on summer. She told it took more than 2 hours, just one
Jesus.
lobster. So how much time it is taking for you guys?
Roughly
Uh,
an hour, maybe just
Hour?
over.
yeah, it just
Hour is
depends.
fine, I think.
Yeah.
Uh,
But the monotone like talking is what gets me. That's where I have to really focus. That's what makes it seem like longer than an hour is that there is no change in tone. It is simply like Bueller, Bueller.
Yep. It's so boring. Go grab a box of gloves. Now move across. I'm like, come on, man.
Thank you for getting my peach juice. Now I can sell more peach juice. You get a year supply. Thank you for doing the simulation.
I'm like, if this wasn't fourth grade, I would not be doing it.
I feel that.
I don't know.
Oh, but it's fun.
Yeah, so last spring, when we had to shut down, I think it's on February, right? So lots of people jumped into Labster. I was the only person. I told no. My student, they are not going to work on Labster. So what I did, I made all the videos and all the lab I just made in by my way, and we didn't do lobster um, on spring, but summer I really didn't have any choice. So somebody else actually taught the lab part. So he told, no, I preferred lobster. So lobster.
Oh,
This
really?
semester we have to, divide the class into two groups. So we
I
really
know that they
didn't
were
have
saying.
choice.
I
So
I know that I had a family member who was doing like a chem class online, and whenever he was doing his labs, he was literally being sent a box every week for him to do his lab, and he'd have to take pictures throughout what he was doing and send it to his professor. But on the flip side of that, with how hard that this is coming from a person who's recently interviewed for Amazon, and he turned it down due to the fact that a lot of Amazon workers like had a mad breakout of COVID. Like I don't know what happened, but Amazon facilities and anything dealing with a package apparently got like pelted with this stuff.
hmm. I'm thinking like next semester. We had meeting today, and I think we will have the same procedure next semester spring. Like
Yeah.
micro should be hybrid. So I'm not sure about Labstar. So I'm thinking what to do, how to
Right.
replace
I'm thinking it's going
Labstar.
to continue until about summer at least.
Hmm.
There's a vaccine.
What's the time? Let's
Time to
start.
start.
Oh, yeah. Okay.
Oh,
So
my
I
bad.
think I stopped over here, right? The classification.
Yeah.
Yeah, and I talked about it, this slide.
Yeah, I think so.
Okay. So let me just go very quickly again. Um, so basically, we have gram-positive bacteria, gram-negative bacteria based on the cell wall composition, right? So now gram-positive bacteria, they are subdivided into two groups, low G plus C and high G plus C. Okay, I will talk about it later. But these are the two groups under gram-positive bacteria. Now, gram-negative bacteria, they are subdivided into two groups, proteobacteria, non-proteobacteria. Now, proteobacteria, they are again subdivided into 5 groups, alpha, beta, gamma, delta, and epsilon. Non-proteobacteria, they are subdivided into 3 groups, Spirochetes, CFB group. CFB group, actually 3 different groups together, they make CFB group. And the Planktomycetes. So these 3 are under non-proteobacteria. Now there are phototrophic bacteria. Phototrophic bacteria, it could be both gram-positive and gram-negative bacteria. The group of bacteria who actually does photosynthesis. So basically photo means light. So they absorb sunlight, and from the sunlight, they get, by utilizing the energy of sunlight, they actually make their own food. That's why that's a trophic, food trophic. Okay, so that phototrophic bacteria, they are both gram-positive and gram-negative bacteria. So that's the whole classification. So our first group, the proteobacteria, means we are going to work on the gram-negative bacteria. Okay, so gram-negative bacteria, it's a very large group, huge number of bacteria. They are much larger than the gram-positive bacteria. So the first group, the proteobacteria, basically the scientist Carl Woese, he first not discovered the bacteria were there, but he identified the similarities among all these purple bacteria and their relatives. Okay, so he found out a large group of bacteria by RNA sequencing. They showed similarities. Based on that RNA sequencing, he suggested that this group of bacteria must be under one phylum or under one group. And from that time onwards, actually we grouped all these purple bacteria and just close relative of purple bacteria under this proteobacteria. Previously, we used to use that name, that Carlu's name, the purple bacteria, but then finally, ultimately, we gave the name of proteobacteria. We actually chose this name from Greek god Proteus, who could assume many shapes. So basically, because of the huge diversification, we gave this name, the proteobacteria. So proteobacteria, under proteobacteria, our first subgroup is alpha proteobacteria. So this alpha proteobacteria, the large number of bacteria here, they are called oligotrophs. Okay, so the word oligotrophs means the group of bacteria who can grow at very low level of nutrients, or you can say that they require very minimum amount of nutrients, and that's why they are called oligotrophs. So this group of bacteria, they can survive at the bottom of the ocean, okay, ocean sediments, glacial ice, where the nutrients, number of nutrients or amount of nutrients are very poor. This group of bacteria, they can grow over there, and that's why they are called oligotrophs. Okay. There are two very important taxa under alpha proteobacteria. One is Chlamydia, the another one is Rickettsia. Now these two, both of these bacteria are called atypical bacteria. Atypical bacteria. Why atypical? Because like Chlamydia, they don't have any cell wall. So Chlamydia, no cell wall. Okay, they don't have cell wall. So this is the only group of bacteria you can understand that they don't have cell wall. Rickettsia, they are so small, very minute, and this group of bacteria, mostly almost not almost all alpha proteobacteria, but most of the alpha proteobacteria, it's very difficult to observe them under microscope, bright field microscope inside the lab. It's very difficult to grow them in lab because of all these characteristics, like they don't have cell wall. They are so small, you cannot stain them. Okay, so you cannot give them any color. Okay, so many different type of staining techniques are there. Even now, not only the gram stain or acid fast stain, we have so many different types of staining procedure, different mechanism, but still it's very difficult to stain this group of microbes, and as a result you really cannot observe them under bright field microscope. Okay, so that's the reason. So basically they are not like other bacteria, so that's why they are called atypical bacteria. Okay. These two taxa also are called obligate intracellular. Okay, so one thing, all the red words, in my PowerPoint are very, very important. Make sure to know all the definitions. Okay. So over here, the obligate intracellular pathogen. So obligate means you can understand that must. Intracellular, they are actually they can grow inside a living cell. Okay, they can multiply, they can do all the metabolic activity inside a living cell, okay, of a host's living cell. But outside of host cell, means in the air, they really do not, they don't have any ability to do any metabolic activity. Basically, they are metabolically inactive outside of a host cell. So to survive, to multiply, to divide, to grow, they must have a living cell. It's almost like virus, you know. They're different because virus, we really don't know that they are living or non-living, but um, this group of bacteria, they're living, that for sure. But just like virus, they must have host cell or living tissue if you want to culture them inside a lab. And that's the reason they are very difficult to grow in lab. And that's the reason we know very little about them. So you really need to supply the living host cell. Like virus, how we actually cultivate virus? In most of the time inside a turkey or chicken egg, okay, chicken or turkey's egg we use to grow virus in lab. Chlamydia rickettsia, we have to have, these are mostly work on the, you know, the animal tissue. So we must need to supply living animal tissues to grow, you know, all these bacteria. And that's the reason they are called obligate intracellular pathogen. So remember that outside of living host, they don't have any metabolic activity. They behave like a dead cell. Okay. Now the diseases, this chapter is going to talk about so many diseases, you know, like this chapter is pretty easy to understand, but the problem is you have to remember so many information like the disease, name of the disease, sometimes the symptoms, and the pathogen. Okay, so you really need to put a little bit more time to memorize all those. So over here, that rickettsia, that's the name of the bacteria. These bacteria infect both the tick and the human. Okay, so if you get a tick bite, okay, or if you get a bite from infected tick, then you are going to get that Rocky Mountain spotted fever. Another species of same genus, that Rickettsia prowazekii, anybody can get infection from the lice bite of lice. Okay, it's called epidemic typhus. What happens in this case? These bacteria actually they actually infect the endothelium cells. Endothelium cells. Endo means inner. You can understand that. So basically endothelium means the lining. Okay, so basically inner lining of the blood vessels. So basically what they do, they infect the inner lining of your artery, of your vein. Okay, so very close to the blood, actually. That's what I want to tell. Very easy access to your blood circulation. So they infect that endothelium cells. Okay, so that epidemic typhus, what happens, like high fever, abdominal pain. Okay, these are the symptoms, sometimes delirium. So basically it starts working with the brain, and the person sometimes forgets even his or her name. So all those things happens from the epidemic typhus. Now that rickettsia, that Rocky Mountain spotted fever, it's actually one type of meningoencephalitis. Okay, a meningoencephalitis means meninges. I think you all know the meninges, that actually 3 layers we have surrounding our nervous system, like our brain and the spinal cord. Okay, outside that's the protective barrier of our nervous system. So this disease, that Rocky Mountain spotted fever, actually attack that meninges. Okay, so swelling, basically that meninges get swelled. So basically outside of the brain and the spinal cord, that layer gets swelled, and lots of pain, high fever, and all those things, all the symptoms arises from that, and accumulation of lots of water in between the meninges. So that's the Rocky Mountain spotted fever. Then another 2 species, or you can say another 2 taxa, are the Brucella and Agrobacterium under alpha proteobacteria. So Brucella, it comes from the infected cow. So basically infected cow milk, you can say. So if that cow is infected with the bacteria, then we are going to get the disease, and it's called brucellosis. Okay, the name of the disease, brucellosis. Agrobacterium, it's the plant disease. Okay, basically these bacteria infect the plant. Okay, so these are few examples of alpha proteobacteria. Just a second. And we talked about rickettsia, now the chlamydia. So chlamydia, what is happening? Okay, so the chlamydia, another alpha proteobacteria. Remember I told that they don't have any cell wall. Okay, so no cell wall. This bacteria, usually they move from one person to another person, or you can say that inside the environment, they live or they survive as elementary bodies. Okay, it's a kind of endospore kind of structure, spore, you can say. So basically just under a plasma membrane, they don't have cell wall, but they do have plasma membrane, cell membrane. They just protect the DNA, genetic material. Okay, and they're not exactly the spore, but they behave like a spore, and that's the reason instead of calling them as endospore, we call them elementary bodies. Okay, so these elementary bodies, they actually float, within the environment, but once they get chance to enter inside the epithelial cells, you know the epithelial cells, right? All of the covering of our body are made up with epithelial cells. So once they enter into that epithelial cells, what they do, they start utilizing our ATP, okay, from that epithelial cells. So basically they use our energy, our protein and all those things, and they convert themselves into reticulate bodies. Okay, so basically the active form of chlamydia is now called the reticulate bodies. They can multiply. So elementary bodies, they cannot actively divide. Okay, to divide or to multiply that elementary bodies, they need a little bit of modifications, and they convert themselves into reticulate bodies. These reticulate bodies, they multiply, they divide, and they utilize the cell's energy, food, everything, and ultimately they completely occupy the cell, and finally they lyse the cell. And when they lyse the cell, then they leave that epithelial cell. So when they leave, the reticulate bodies we can tell them again as elementary bodies. Okay, so somebody is joining, and I don't know, I'm admitting, but why can't see? Okay. So that's the whole life cycle of chlamydia. So when they are outside of a living cell, they remain as a spore-like structure, which is called elementary body. Once they enter inside our cell, epithelial cell, they start utilizing the ATP and other material, and when they're inside a cell, they behave, they actually have the ability to do metabolism, right? So then we called all those elementary bodies as reticulate bodies. Now reticulate bodies, they utilize all of our energy, ATP and all the materials needed to divide, needed to multiply, okay? And when they absorb everything from the cells, nothing left inside the cell, then they leave the cell, they lyse the cell, and they basically they comes out from the cell again in the form of elementary bodies. So that's the basically two form, elementary bodies and the reticulate bodies. So that's the life cycle of chlamydia. And remember, as I told, that when they're outside, they behave like a dead cell. That LGB's chlamydia trachomatis, highly pathogenic, contagious, very dangerous bacteria. They're responsible for lymphogranuloma venerum. It's a sexually transmitted disease that infect the lymph nodes. Very, 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 very common in college campus, very highly contagious. So that's the chlamydia trachomatis. Same bacteria, same species, that chlamydia trachomatis, they are responsible for the trachoma, that retina that actually affect your retina inside your eyes, and ultimately, it could bring the blindness. There is no treatment, still, very little treatment, because remember I told that we really have very little study about all this, that chlamydia and rickettsia, because it's very difficult to culture, to grow, and to observe, to study. Okay? And at the same time, both the genus are highly contagious. Okay, so that's another problem to study. Our next group, who is waiting? Okay, nobody. Our next group is beta proteobacteria, another group. So alpha proteobacteria, our previous group, they were oligotrophs. Oligotrophs means they need very little amount of nutrients, not much. But this group, these are called eutrophs, okay? Because they need a copious amounts of nutrients. They are very fastidious, they have special needs, there are few bacteria who need high amount of, you can say, the nitrogen. There are other fastidious bacteria who need lysed RBC. Some organisms, they need phosphorus. So basically, based on the bacteria, they have special requirements. Basically, if you don't supply that requirements, then you cannot grow them. Okay, it's very difficult to culture all those bacteria, and that's the reason they are called eutrophs. Not only the number of nutrients, they need a huge amount. So both the number and the amounts of nutrients actually matter in this case. Okay, they are not like oligotrophs. So you cannot see all these bacteria everywhere, okay, because they are fastidious. Fastidious means they have special requirements, okay, like Neisseria gonorrhea. So this bacteria, that Neisseria, the genus Neisseria, they are very, very fastidious. They, you only can grow this Neisseria genus with chocolate agar plant. Okay, it's chocolate agar. That does not mean that we really add the chocolate, but it's a lysed red blood cell. So basically, to grow this bacteria, we must have supply of blood. So we cannot supply the human blood, but what we do, we actually supply 5% sheep blood, and all the RBC should be lysed RBC. Okay, it's not like regular blood agar. Okay, this blood has to be lysed. So that's a special requirements. Okay, if we cannot supply this lysed RBC blood agar plate, then we cannot grow this bacteria, this genus, the Neisseria. Neisseria gonorrhoeae, responsible for gonorrhea, sexually transmitted disease. Another one species, the Neisseria meningitidis, responsible for meningitis. Again, meningitis is the swelling of the meninges outside of the skull. Another important species is Bordetella pertussis, responsible for pertussis or a whooping cough. What is happening with Christina? I'm constantly accepting, but I don't know, something is going on. I think she is trying to join, but she cannot. Anyway, so the Bordetella, responsible for pertussis, whooping cough. In this case, what happens, I hope you know, like all of our inside passageway, like inside your digestive system, your respiratory system, all the internal body tubes must have cilia. Okay, that cilia and also the mucus cell, mucus membrane, you can say. That mucus and cilia actually helps to, you know, it brings the smooth passage of anything, like if it's food, that movement of cilia and the mucus actually help food to move through. Exactly same way your air tube, your respiratory tube, that beating of cilia actually helps the air or oxygen to go inside. In this case, the Bordetella pertussis, this bacteria, what it does, it actually paralyzed the cilia inside of the respiratory tract. Okay, and as a result, air, that sufficient amount of oxygen cannot reach inside the lungs, and ultimately not inside your blood. So that creates a huge cough with a bad sound, you know, and that's why the name is like whooping cough, because of the coughing sound. So that's the pertussis disease. It's, if you don't treat at the beginning, then it's very lethal. Next group, Gemma. Gemma, it's a huge, the largest group of proteobacteria, you can say. Okay, largest class. One important genus under this Gamma proteobacteria is Pseudomonas. Okay, the family Pseudomonaceae. So under Pseudomonaceae, we have that Pseudomonas. One of the species is Pseudomonas aeruginosa, responsible for urine infection, responsible for respiratory disease, for, you can say, cystic fibrosis patient. Okay. Then if they are responsible for burn and cut infection, okay, and it's very difficult to treat. That swimmer's ear infection, that is caused by this Pseudomonas. Okay, swimmer's ear, or swimmer's ear disease, that same bacteria. there is at this moment, it's very difficult to kill Pseudomonas with the antibiotic, okay? Most of the antibiotic does not work because this group of bacteria can form biofilm very quickly. Okay, they can attract other bacteria to form that biofilm. It's very important bacteria for the biofilm. And remember, I told that when it is a biofilm, we don't know how to treat, because lots of different groups of bacteria, protozoa, viruses, fungi, together, all these different types of microbes together, they just behave like one single microbe. Okay, so basically, we don't know the identity when they make biofilm. Okay, so that's the very hardest part to treat in biofilm, and no antibiotic actually works. So Pseudomonas is one of the bacteria who are responsible for that biofilm. Very important. I can give you the question like, which bacteria you can find inside a biofilm? So Pseudomonas. Till now, you just studied this bacteria. Another genus, Vibrio cholerae, responsible for diarrhea. Basically, they actually attack the digestive system, large intestine. So a large intestine actually absorb too much water from the body, and the body gets dehydrated, and the stool becomes really watery. So that's the Vibrio cholerae. There is another bacteria, Alivibrio fischeri. This is one bioluminescent bacteria. We have lots of bacteria who can do bioluminescence. Means in dark, they can create. They can basically they can. You can say that create light inside their body. So these bacteria, they have a mutualistic relationship with one type of squid. It's called Hawaiian bobtail. These two are actually they do mutualism. Okay. So because of the light, the predators, they don't attack the squid, and but the Alivibrio fischeri, it gets a place to stay and the food from the squid. So that's how they do their mutualistic relationship. Okay, so the interesting fact is that this bacteria is a bioluminescent bacteria. Legionella, another group, I hope you have heard about that. This is the Legionnaire's disease. It occurs inside the AC unit or heater unit, inside a building where there is a little warm water, and that circulates through the building. So they actually need that settings, that temperature. So the causative agent of Legionnaire's disease thrives in the warm water. Outbreaks of Legionnaire's disease often originate in the air conditioning units of large buildings when water in or near the system becomes contaminated with Legionnaire pneumophila. So Proteus, another genus, they are responsible for sepsis, different type of sepsis, Klebsiella. We usually work with these two bacteria in lab, but this semester you are not going to work on this. But Klebsiella, they are responsible for one special type of pneumonia. Enterobacter, they are responsible for different type of enteric disease. Serratia, they are actually you can say that they're not that pathogenic for human. I'm talking about Serratia in room temperature. You can see in lab, they actually make red pigment, okay, in room temperature. So that inside your bathroom shower area, you will see that tiles become reddish a little bit. So that's the because of the accumulation of Serratia. Okay, Serratia, they are not that pathogenic, but they are actually called opportunistic bacteria. Or you can say they are opportunistic pathogen. So opportunistic pathogen, this word means usually they are not pathogenic to human. Okay, but if they get any chance, if there is a cut or burn, or somehow if they get entry inside the blood circulatory system inside your body, then they are pathogenic, but otherwise they are not pathogenic. Okay, so yeah, on your shower floor, you can see that red pigment, but that's usually not pathogenic. But if you have any cut, and if you stand, if your cut get contact with that Serratia, then you might have problem with this bacteria. Otherwise, they are not that harmful. So that's why they are called opportunistic pathogen. One group of bacteria who are under this group, that gamma protibacteria, this group is very, very important, a highly pathogenic group, very large group. There are so many bacteria who are highly pathogenic, highly contagious, and at the same time who are very helpful. We use them for so many purposes. Okay. So one group of bacteria under this group is enteric bacteria. The word enteric means they live inside your intestine. Okay, so inside your, basically they are the gut bacteria. Okay, not all enteric bacteria are pathogenic. On the other hand, not all enteric bacteria are helpful. We have both inside our gut, the pathogenic bacteria and the helpful bacteria. Anyway, this enteric bacteria, you can understand that they remains inside the gut, so they are facultative anaerobe. Okay. So the word, the facultative anaerobe. Facultative anaerobe means basically they are actually aerobic bacteria, means they must have oxygen supply, okay, for their respiration. They are aerobic bacteria, okay, but they're not obligate aerobe. If there is no oxygen, they can survive because they can do anaerobic respiration, okay, when the environment is not favorable. So that's the reason they are called facultative anaerobe. So remember, this terminology are very, very important. Facultative anaerobe means they are basically aerobe. They must have oxygen supply, not must, but they need oxygen. They usually do aerobic respiration, just like us. For our respiration, we use oxygen every time, but for us, we are obligate aerobe. Like without oxygen, we cannot survive. But this group of bacteria, enteric bacteria, when they are outside of the gut, they are actually aerobic bacteria. But without oxygen, they can survive. They change their metabolism, So that's the adaptability, resilience, you can say, of these bacteria. So they change their metabolism and they convert themselves into facultative anaerobe. Okay, I hope you got the term. They ferment carbohydrates. So that's the enteric bacteria. Okay, so all the gut bacteria are called enteric bacteria. Now these gut bacteria, they are subdivided into two groups. One is coliforms and another is non-coliforms. Okay, coliform, this group of bacteria, they ferment lactose. Okay, they ferment lactose and they produce acid and gas, lactic acid and gas. Okay, so that's the coliform bacteria, like E. coli. They are coliform bacteria. But on the other hand, non-coliform bacteria like Salmonella, that Salmonella is responsible for the typhoid fever. That Salmonella, they're non-coliform. Non-coliform, they may or may not ferment lactose. Okay, but if they ferment lactose, they cannot produce acid and gas. So usually non-coliform, they cannot ferment lactose. But if they ferment lactose, then they don't produce acid and gas. So basically that lactose fermentation is not complete for non-coliform bacteria. Usually they don't ferment lactose. But if they ferment, that's an incomplete fermentation of lactose, and as a result, they cannot produce acid and gas. Okay, so that's the difference between coliform and non-coliform. I think inside your book, it says that coliform, they ferment lactose, non-coliform, they don't ferment lactose. No, that's not true. Non-coliform, usually they don't ferment lactose, but they have ability to ferment lactose partially or incompletely. Okay, so that's the definition of non-coliform. They may or may not ferment, but this group of bacteria, they produce acid and gas from the lactose fermentation. This group of bacteria, they don't produce acid and gas because it's a incomplete fermentation of lactose. Example, Salmonella. These two are completely opposite, you know, the Salmonella and Escherichia coli. The next one, Delta Proteobacteria, very small group of bacteria. They are mostly sulfate-reducing bacteria. Sulfate-reducing bacteria, or SRB group, is, remember I told that because of our respiration, we must have oxygen. Okay, oxygen. Without oxygen, we cannot break down the carbohydrate. This group of bacteria, that sulfate-reducing bacteria, they actually reduce the sulfate. Okay, sulfur product. They really do not need any oxygen. Example, Desulfo Vibrio oral, they are responsible periodontal disease, gum disease. Another one group, Myxobacteria, they're the soil bacteria. Recently, we are working on it to produce, you know, different types of medicine. Lots of medicine we actually make from this group of soil bacteria. This group of bacteria, they're very interesting. They make fruiting bodies. Just like previously we had idea that that's the plant. Previously we used to think on that way, but now we know that they are not the plant, they are animal. So these bacteria, they actually can communicate with other group of bacteria. They know how to communicate with other group of bacteria. Very, very social organism, this group of bacteria. So usually non-pathogenic. Most of them are. It's a large group of bacteria. Very helpful. Epsilon Proteobacteria, Helicobacter pylori. You know, our dangerous enemy, responsible for gastric ulcer, responsible for cancer, intestinal cancer, colon cancer. Very, very dangerous, this group. Who is joining? I don't know. One example, Campylobacter, Campylobacter jejuni, actually. It's called Jejuni. So this is the species Jejuni. Um, Responsible for food poisoning, it occurs mostly from the chicken. So basically, if you go to a restaurant and if you ask um, to cook, they ask you, okay, what type of cooking you want. So you really need to tell that well cooked. Okay, so if it is medium or low, then the chances are there that you are going to end up with Campylobacter. Okay, so diarrhea, blood. It's not necessarily it's bloody diarrhea, but it's a diarrhea, highly pathogenic and contagious. Helicobacter pylori, I told you that they're responsible for gastritis, ulcers, intestinal cancer. Okay. Previously, we really did not have any idea about gastritis. We used to think that gastritis because of the acidity. But now we find out that this group of bacteria, they have one enzyme, it called urease. That enzyme, so if this is your intestine, you can understand that our stomach, it secretes hydrochloric acid, right, HCl. Okay, so that high acid can destroy your intestinal wall, right? So to protect your intestinal wall from hydrochloric acid, what we have a special layer, mucus layer, outside of that intestinal wall. It's a mucin layer. Uh, Now, this urease enzyme of this group of bacteria, what they can do, they actually, this urease can make it liquid, basically, this layer, okay? They make it liquid, and they make a hole. As a result, they can enter inside the intestinal wall. So because of the liquidity, of that mucin layer, because of the presence of urease enzyme, they can have direct contact with the intestine wall, and they can make a hole over here. So recently, I think last 10-12 years, we came to know that Helicobacter is responsible for gastritis. Now we treat gastritis patients with antibiotic, but previously we really did not have idea. So gastritis patient we used to treat with like antacid medications and all those. So last 10-12 years we came to know about this bacteria, and they are responsible for colon cancer and all those. Spirochetes, so that's the first group of non-proteobacteria. They, from the name you can understand they're spiral shaped, or you can say corkscrew shaped bacteria. Highly motile, they have special type of flagella, okay? And this flagella, it's called periplasmic flagella. So basically, these flagella, they rotate this flagella with high speed, but this flagella remain inside the periplasmic space. Okay, in between the inner membrane and the outer membrane. Okay, so there is a jelly-like area, semi-solid area. So that flagella, periplasmic flagella, remains inside the periplasmic space. They rotate vigorously, and that's how this bacteria move so quickly. One example, Treponema pallidum, responsible for syphilis, another sexually transmitted disease. Another one, Borrelia burgdorferi, responsible for Lyme disease. Lyme disease, you can get it from if you get a bite from tick. You can get it from tick bite. Okay, so you can get it from infected tick. So rash, high fever. Okay, I forgot the name of the rash. There, I think bull's eye or something. It has special name, special type of rash you can get. So that's a Lyme disease. Lyme disease. If you don't treat properly, sometimes you can get that arthritis. Okay, from this Lyme disease. So very, very important, these two, Treponema pallidum for the syphilis and the Borrelia burgdorferi for the Lyme disease. But this chapter, make sure to remember, okay, so make a chart. Okay, so for syphilis, name of the pathogen, Treponema pallidum. So you have to really memorize so many name of the pathogen, the disease corresponding, the disease, and few cases, the symptoms. Then the next group of non-proteobacteria, the Cytophaga, Fusobacterium, and Bacteroids. These three groups, previously they were under non-proteobacteria, but they were in different groups. But because of the DNA sequences, similarities in DNA sequences, now we are keeping all three together, and that's why this group is called CAB group. Okay, together, Cytophaga, Fusobacterium, and Bacteroids. See similarities in the sequence of nucleotides in DNA. Rod-shaped bacteria, they are anaerobes. Anaerobes means they really don't need oxygen, able to ferment cellulose in rumen. Rumen animals, you can find out inside their stomach. And so even like whatever the salad thing you eat, like whatever the cellulose you eat, we really do not have any enzyme inside our body that can ferment cellulose. Okay, to ferment cellulose, like when you eat raw spinach as a salad, to digest that spinach, you must have this group of bacteria, the bacteroids, inside your gut. Without this group of bacteria, we cannot digest cellulose. Okay, it's not only us, not only the human, all rumen bacteria. Next, the first group, the Cytophaga, they are motile and aquatic. Fusobacterium, they are responsible for your gum, periodontal disease. Now these group bacteroids, they are very, 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 very important bacteria. This group of bacteria, they comprise up to 30% of the normal microbiota in the human gut. Okay, so the word microbiota. Microbiota means a group of microbes you can find in an environment, in a specific environment, like if it is your mouth. Okay, so the mouth microbiota, usually what are the regular bacteria you can find inside your mouth, that's your mouth microbiota. That will not match with your skin microbiota. Okay, so microbiota means a group of organism or microbes, you can say, in a specific environment. That's the microbiota. All microbes together makes that microbiota. Now the microbiota, they are two different types. One is resident microbiota, and another group is transient microbiota. So resident microbiota like Fusobacterium. Every time you can find them inside your mouth, or you can say the bacteroids inside your gut. Every time you can find them. So this group, that resident microbiota, they are constantly, you can say, they are constantly living in or on your body, like Staphylococcus epidermidis, always they are on your skin. Bacteroids, they are inside your body. Okay, so bacteroids, they are our normal gut microbiota for human. Staphylococcus epidermidis, they are our resident microbiota for our skin. Now the transient microbiota means occasionally you can say occasionally you can find them. Okay, so if you have any infection like now the coronavirus, so that would be your transient microbiota. So because of the disease they are there, it might not be the disease, like sometimes like Streptococcus pyogenes, not necessarily they are every time pathogenic. Sometimes you can find that Streptococcus pyogenes inside your mouth, but not necessarily they make you sick. Okay, not all the time, because they are opportunistic pathogen. So so you can find them occasionally. So they are the transient microbiota. Okay, so these are the two different types of microbiota. Now these bacteroids protect human from secondary infection. Okay, so what happens, this group of bacteria inside your gut, they don't allow any other bacteria to grow inside the gut. Okay, because they produce so many different type of metabolic products. Those products are not favorable for other group of bacteria. Okay, so if you have enough amount of bacteroids, you can easily avoid the secondary infection. Okay, so they actually protect us from other pathogenic bacteria. And as I told that they digest cellulose. So this group of bacteria, they're basically very, very helpful bacteria. Now the phototrophic bacteria, they do photosynthesis. Okay, so they do photosynthesis. Now two different types. It could be oxygenic photosynthesis or non-oxygenic photosynthesis. Okay, so oxygenic photosynthesis means this group of bacteria, when they do photosynthesis, they really need the presence of oxygen. Oxygen is necessary for this group of bacteria. Okay, but non-oxygenic, they really do not need oxygen. Okay, they use something else for their photosynthesis. Basically, they use organic or inorganic sulfate or sulfate product or malic acids, and they really do not produce oxygen. So but oxygenic, they produce oxygen, like plant, they do oxygenic photosynthesis. They do oxygenic photosynthesis, and that's the reason we survive, because for our metabolism we must need oxygen. Okay, so basically this group, oxygenic bacteria, what they do, they use carbon dioxide, sunlight to produce oxygen. This group, they, instead of carbon dioxide, they use something else, and they don't produce oxygen. So both their photosynthesis, this group, both proteobacteria and non-proteobacteria, oxygenic and non-oxygenic photosynthesis. One group use bacteriochlorophyll, blue, purple, or green, or carotenoids, red or orange. So to do photosynthesis, you know that, okay, so it should be one. You know that for photosynthesis, the plant use chlorophyll, right? One group of bacteria, they use that chlorophyll, or you can say it's plant chlorophyll, like chlorophyll A, chlorophyll B, phytokines and all those. But this non-oxygenic, they have that bacteriochlorophyll. It's not exactly the plant chlorophyll, but some pigment that can absorb photon, just like chlorophyll. Okay, but they are only for these bacteria. So that's why they are the bacteriochlorophyll. They have blue, purple, or green, mostly the blue, purple, and green bacteriochlorophyll. Some of them has carotenoids that gives that red and orange color. Now, this slide actually mostly talks about the non-oxygenic phototropic bacteria. So this non-oxygenic phototropic bacteria, so they really do not produce oxygen. Okay, they are subdivided into sulfur and non-sulfur. So basically, that non-oxygenic, they are subdivided into sulfur or non-sulfur bacteria. Now, the sulfur bacteria, they usually use sulfide or sulfate. So this group use sulfate. Okay, means it's inorganic compound. Okay, so this group, sulfate bacteria, they use inorganic compound, inorganic sulfate, and non-sulfur bacteria, they don't use the sulfate, but it's inorganic. Organic, they use malate kind of stuffs. Okay, so they use malate. So that's the organic. So that's the difference between non-sulfur and sulfur. Sulfur, they are inorganic. They use inorganic compounds. Non-sulfur bacteria, they use organic compounds. Now, the sulfur and non-sulfur both are subdivided into two groups, purple and green. You know what color or pigment they produce based on that name. So purple sulfur bacteria, they oxidize hydrogen sulfide into sulfur and sulfuric acid. Carbon dioxide is the only source of carbon. Very, very important. This slide is very, very important. Make sure you remember the definition. At least one questions you are going to get from these four definitions. So purple sulfur bacteria, very important. They oxidized hydrogen sulfide into sulfur and sulfuric acid. So basically, they breaks down hydrogen sulfide, produce sulfur and sulfuric acid. And the only carbon source is the inorganic carbon dioxide. Green sulfur bacteria, they use sulfide, mostly the hydrogen sulfide. Purple non-sulfur bacteria, they use hydrogen. Remember, I told that non-sulfur means organic. Or whenever you see the word organic, when it is organic, two compounds must be associated with each other. That's a carbon and hydrogen. Okay, that represent when carbon and hydrogen together associated with each other, then that represents the organic. Okay? So purple non-sulfur means they're organic, so they use hydrogen. Green sulfur is substrates other than the sulfides because as I told that they use the organic form. Make sure you remember this. Now another group of bacteria who are actually oxygenic, like cyanobacteria. Okay, so oxygenic bacteria means they produce oxygen. Okay, just like plant. And remember, I told that um, enter Amazon rainforest, how much oxygen they produce. More than that oxygen we can get from the cyanobacteria. So they are the oxygenic phototrophic bacteria. And they have that chlorophyll like plant, like chlorophyll A, phycocyanin, cyanophycin, chlorophyll B, exactly like plant. Microcystis aeruginosa is a type of cyanobacteria. They actually produce that it's kind of algal bloom kind of stuffs. Okay, very poisonous neurotoxin that affect our nervous system, resulting in blooms that are harmful to fish and other aquatic animals. So that's the only cyanobacteria. Basically, all of the cyanobacteria are very helpful. This is one example who are not pathogenic but not helpful. They are harmful to the aquatic organism, aquatic animals. Now we are on gram-positive bacteria. So G plus C. So gram-positive bacteria, high G plus C and low G plus C. So basically, if the person, I hope you remember from the DNA nucleotide structure, there are adenine, guanine. I hope you remember that adenine, guanine, cytosine. So that guanine and cytosine, the percent of guanine and cytosine. So I hope, okay, so let me go into the detail. So I hope you remember that T goes with A and G goes with C, right? Okay, so the percentage of these two and the percentage of these two together makes it 100%, right? And percentage of G equals percentage of C. Over here, percentage of T equals percentage of A. That's how it goes, right? So high G plus C means guanine and cytosine percentage is more than 50%. Okay, so higher than the T and A. So that's the high G plus C, and low G plus C, they have less than 50% guanine cytosine. So high G plus C gram-positive bacteria, mostly we are going to work on the actinobacteria, a huge group of bacteria. We get the antibiotics from this group. They are the soil bacteria, mostly aerobic. A few of them, they remains inside the mouth, and they're responsible for periodontitis. Mycobacterium, one bacteria, that Mycobacterium tuberculosis and Mycobacterium leprosi, they are acid-fast bacteria. They have mycolic acid inside their cell wall. Another bacteria, Corynebacterium diphtheria, causes the deadly disease diphtheria. What happens? This bacteria actually creates a pseudomembrane inside your respiratory system, and because of the pseudomembrane, person cannot breathe. It blocks the passageway. Okay, so that's the Corynebacterium diphtheria. Gardnerella vaginalis causes the bacterial vaginosis in women. So this high G plus, it's a huge group of bacteria. The soil bacteria, Actinomyces, large group of bacteria. We get antibiotic. Then these few are actually harmful bacteria. Low G plus C, the Clostridium difficile, gram-positive broad-shaped bacteria cause severe colitis and diarrhea. Often after the normal gut microbiota is eradicated by antibiotics produced in those spores. Clostridium all the canned food, most of the time that food poisoning from the canned food occurs because of this group of bacteria, the Clostridium, mostly the Clostridium perfringens. Food poisoning, especially in the canned food. Clostridium tetani, they secrete neurotoxin. They are responsible for spastic paralysis. So basically, they inhibit the nerve impulse. Okay, so that impulse, that message, or the information moves through the neuron, right? These neurotoxin actually inhibit that impulse, that movement of information. Another one, Clostridium botulinum, they're responsible for botulism. They are responsible for flaccid paralysis. In this case, what happened that they actually block the end portion of the neuron, terminator. Basically, they stop secreting acetylcholine. So as a result, the information cannot move from one neurons to another neurons. So that's the botulism. Another gram-positive bacteria, Lactobacillus, Enterococcus, Streptococcus. One bacteria, the Streptococcus pyogenes. Anything with word strepto means chain. Okay, it's a long chain of coccus bacteria, or round-shaped bacteria, that Streptococcus pyogenes responsible for impetigo and tissue necrotic diseases. Over here, they are beta hemolytic. Okay, you can see they completely lysed the RBC. So basically, this group of bacteria, these bacteria, the Streptococcus pyogenes, they can lyse the RBC. They can break down RBC. Okay, red blood cell, and inside the blood agar plate, you can see that these bacteria, when they grow on blood agar plate, and they completely lyse the RBC, completely destroy, or you can say completely. Basically, they eat the RBC, and as a result, the agar becomes completely transparent. So that's the beta hemolytic bacteria. Bacilli, rod-shaped bacteria. So over here, Bacillus cereus. So bacilli could be both gram-positive or gram-negative, like B sub, Bacillus cereus or Bacillus subtilis. They are the gram-positive. Rod-shaped bacteria, Escherichia coli, that's a gram-negative bacteria. Now we are actually talking about the shape. Bacillus in anthracis, they are responsible for anthrax disease, Staphylococcus, so the differences between Streptococcus and Staphylococcus. Staphylococcus, you can see both are coccus, rod-shaped bacteria. Staphylo means they are in a cluster. Okay, they mix a cluster. So most of the time it looks like a grape-shaped cluster for the Staphylococcus. Strepto means it's a long chain. This group, Staphylococcus aureus, facultative anaerobe, and again the facultative anaerobe means they are actually aerobe, but they can do anaerobic respiration. They are halophilic, non-motile, halophilic. Halo means salt, so they are salt lover. Means high concentration salt, they can survive. Staphylococcus epidermidis, opportunistic pathogen, they reside on your skin. Staphylococcus aureus, they secrete one special type of enterotoxin. All the staph infections, carbuncles, impetigo, cellulitis, these bacteria are responsible. MRSA and VRSA, methicillin resistant and vancomycin resistant Staphylococcus aureus. I hope you all know, like MRSA, mostly you can get it from the gene. We really don't have even we now for the VRSA we really don't have any antibiotic to treat. Evolution of bacteria. So from the tree, the root represent, you can see over here, the Lusa, that's the common ancestor. Okay, so that's the common ancestor of all of us, like we are here, eukarya. Okay, these two are the prokaryotes group. Now, over here, so we all originated, and you can see for a long period of time only this group of bacteria were there up to this point. Okay, now over here at this point there was an evolution, and what happened, this group actually divided into two groups. So basically two groups of organisms originated at this point. Okay, now one group on this side, one group on that side. So that, and from here, from this group, so from this ancestor, both archaea and eukaryotes, means we human originated. Okay, now for the bacteria, our most recent bacteria who are acetothermous, they are also called deeply branching bacteria. They are thermophilic. Thermophilic means they can tolerate high temperature. You can understand, like this period over there on Earth, it was highly high temperature, aquatic, mostly aquatic, high acidic environment was there, right, at this moment almost. So when you can see the eukaryotic, or before eukaryotic organism starts, the Earth environment was like that. Thermophilic, their acidophilus, means high acidic condition they can tolerate, almost 95 degrees centigrade temperature they can tolerate. So this group of bacteria, that acetothermous bacteria, they are called, they are one of the deeply branching bacteria. So deeply branching bacteria, so now how we see the bacteria, they are actually closely represent this group of bacteria, deeply branching bacteria. We have similarities with bacteria with deeply branching bacteria. Before that, they really didn't look like or behave like bacteria. So this group, this deeply branching bacteria, they are the closest ancestor of present bacteria, and they are called deeply branching bacteria. So basically they are, you can say they are the oldest bacterial ancestors, and they are thermophiles, acidophilus. Now the archaea, they are also called extremophiles. Means they live in high temperature, high acid, everything in extreme environment. So Sulfolobas and archaean of the class, Crinarchaeota, they oxidize sulfur and store sulfuric acids in its granules. Okay, Halobacterium, in hot springs you can see that this group of bacteria, they can tolerate more than 120 degrees centigrade temperature. We are actually collecting different types of enzymes for DNA research from this halobacteria. We call halobacteria, but they are actually archaea. Okay, we are not really archaea. They are not pathogenic at all. So that's why in this class we are not going to study in detail about archaea, because they are not human pathogen. Okay, So that's the Archaea. Oh, we are done. Okay, so do you have any question? Anybody? No? No questions?
No, ma'am.
Okay, so I think your test is going to open tomorrow morning, right?
Yes.
Yes, ma'am.
Yeah. And it is going to cover chapter 1, 2, 3, 4.
Yes.
Yes, ma'am.
Okay, and I think it will open till, so tomorrow is Thursday. So Thursday, Friday, Saturday night it is going to close.
So it's open for about 3 days.
Yeah, I think so. Check the syllabus.
Okay.
I think that's how I set it up. So 3 days it will remain open.
And then just to clarify, there is no lab tomorrow, correct?
No lab. This week there is no.
Okay, I just wanted to
Remember,
make sure.
I told you that for this class, one day we are going to set up and the next day we are going to observe. So that's the reason, if one day is holiday, then the next day we cannot do the lab.
Okay.
So no lab, but we are going to start again from next week. Okay?
Y'all have a good night.
Yeah, you too. Be safe and study well. And make sure to work on each and every detail of the text. If you have any questions and concerns, just let me know, okay? Okay, good night.